Noswyth da, good evening, everybody. The warmest of welcomes from here in Cardiff. I'm Roger Whitaker. I'm the Pro-Vice Chancellor for Research, Innovation and Enterprise here at Cardiff University. It's wonderful to have everybody here this evening as part of the wider Cardiff community, including alumni, staff, donors and friends of the university who do such a wonderful job in supporting Cardiff and supporting our mission. Tonight's event will be recorded and you'll be able to receive the link afterwards, and you're more than welcome to share that link with friends, family, colleagues and so forth. This evening, we're taking a look at a really important part of Cardiff University, and we're looking at the journey of how new medicines are developed and how they move through beyond the university. This is a really important area for us here in Cardiff, but it's also a really important area for so many people who are affected by illnesses, diseases and chronic conditions. As we'll all be aware, these are really burdensome with huge direct impact on people and their well-being, but also extending to families, to society and to the healthcare sector. And it's medicines that can really help to alleviate this burden. Yet the path of drug discovery from the laboratory bench through to bedside is really quite tricky. It's notoriously long and difficult. The Medicines Discovery Institute here in Cardiff is a bridge between the basic science developed at the university and the clinical research required to establish the safety and effectiveness of new drugs. I'm absolutely delighted this evening to welcome two colleagues based at the Medical Discovery Institute, the MDI, as we like to say here in Cardiff, and they are Professor John Atack, one of the Centre's joint directors, and Loren Waters, who is a research assistant at the MDI. We've already received some questions in advance, but if you do have questions to ask, indeed you may wish to think about questions to ask, then you can enter them in the Q&A box at the bottom of your screen, and we will answer them as many as we can as time permits this evening. But for now, I'm absolutely delighted to ask John and Loren to take the floor. So welcome and croeso.
Yeah, thanks, Roger. I'll just pull up my slides. Bear with me for a second. Yeah, so thanks for that introduction, Roger. And this is a really very exciting time for the Medicines Discovery Institute. And I'll go into a bit more detail about what we do and what is the Medicines Discovery Institute, a brief overview of the drug discovery process and what we've assembled into our portfolio of the Medicines Discovery Institute, what happens next to our projects. And as I said, this is a very exciting time for the Medicines Discovery Institute, since we have a spin-out company called Drag Therapeutics, which, as we stand now in the beginning of June, will actually come out of stealth mode next week. So that's middle of June 2025. And I'll go into a few details about what Drag Therapeutics is. And what I'd like to emphasise is how the success of the Medicines Discovery Institute is unique within the UK. I think it's safe to say that. And it's unique because of the scientific environment that we have at Cardiff University and the insight of management such as Roger that provides funding that underpins the Medicines Discovery Institute. A lot of universities claim to do drug discovery, but I think that's a bit of a stretch, really. We are full-time drug discovery institute that's embedded within Cardiff University, and I'd like to emphasise that in a bit more detail as I go through the presentation. One of the real privileges of working at the Medicines Discovery Institute within Cardiff University, and we are like a biotech embedded within Cardiff University, is we get the opportunity to work with young colleagues like Loren, early career researchers just starting out on their careers, and hopefully giving them a flavour of what is required for translational science. And by translational science, we mean translating basic scientific knowledge and using their scientific expertise and training to try and make a difference in terms of developing new drugs and new approaches to treatment and translating that basic science into patient impact. And so Loren will give us a brief overview of the project that she's working on with acute myeloid leukemia. So that's a cancer target. The Medicines Discovery Institute is headed by myself and Simon Ward. Importantly, both myself and Simon have an industry background, so we've worked in big pharma. We moved to academia probably just over a decade ago, initially at the University of Sussex, and then more latterly at Cardiff University for reasons that I'll explain in a bit more detail. We have an industry background. We know drug discovery. We are full-time drug discoverers. We have a team involved in commercialization and project management. So that's Richard, Michael, Jenny and Oana. And then we have academics on the left-hand side of the slide here. These are academics based at Cardiff University that are experts in their respective fields. And we help translate their basic knowledge into impact in terms of developing next generation drugs. So this just shows how we have this bench to bedside capability. We at Cardiff University, this is fairly unique. We have some world leading expertise in identifying genes and proteins that are involved in the disease process, whether that's for psychiatrists, so things like schizophrenia or neurodegeneration, things like Alzheimer's disease. We have, again, the world-leading Cardiff University Brain Research Imaging Centre. We plug very nicely in this bench-to-bedside model, taking the basic understanding of the disease process, developing drugs that can then be tested with the cubic brain imaging methodologies to show that a compound gets into the brain and engages the target. And then we have the patient and societal impact. Really important to emphasize that Cardiff University really are world leaders in understanding the pathophysiology of psychiatric disorders and neurodegenerative disorders. And that's just shown in this next slide. So this is the Schizophrenia Working Group, the Psychiatric Genomics Consortium. And the key thing is really, this is a huge global initiative which looks at the genetics of 77,000 individuals with schizophrenia, 250,000 control individuals. And what I'd like to point out here on the right-hand side is the list of all the authors that contribute to this work. This is a huge global consortium, and it's led by James Walters and Mick O'Donovan at Cardiff University. So a really nice example of how Cardiff University are world leaders in understanding the pathophysiology of psychiatric disorders. And the similar occurs for neurodegenerative disorders. So that's the Dementia Research Institute, leading genetics of Alzheimer's disease. There's a very simplified overview of the drug discovery process. So we have somebody sat at a desk, has a bright idea. Let's make some chemicals. And that bright idea is really trying to identify a protein that's involved in a disease process, and therefore modulating the function of that protein with a chemical will hopefully have an effect on that disease process. We then test the compounds, then they go into formulation, stability, and scale up. We submit lots of documentation to the regulatory authorities. Here it's the FDA, the Food and Drug Administration, that's the USA. In the UK, it's the MHRA, or in Europe, it's the EMA. So you put all the paperwork together and ask for permission to test your drug. Can you test your drug initially in healthy volunteers? And that's simply to ask the question, is my drug safe and well tolerated? And if the answer is yes, you test it in phase 2 studies. So these are efficacy studies. So these studies are designed to see if your drug actually has a beneficial effect. Does it have efficacy? And that could be schizophrenia, Alzheimer's disease, cancer. It's the same generic model of phase 1, phase 2. Phase 3 are large clinical studies. And then you put all the paperwork together and ask the regulator, the FDA or the EMA, MHRA, can I have a license to sell my drug? And then hopefully, if you're successful, you can get a drug launched and on the market. So that's a seemingly quite a linear process. The Medicines Discovery Institute is involved in this part here, which is an iterative, making compounds, testing compounds, and then make some more compounds based on the data, test some more compounds. So it's an iterative cycle of hopefully making compounds that are getting better and better. In the real world, I think that drug discovery is more like snakes and ladders. It's a long process. You're winding your way slowly through all these steps. But unlike snakes and ladders, there's no ladders, so there's no shortcuts. There's only snakes, so you can only ever go backwards. You encounter a problem, and that sets you back. And also, unlike snakes and ladders, there are various trapdoors positioned along this pathway from the start to the finish. So you can have a drug that might land on, say, number 94, for example, and that triggers a trapdoor, and your drug just disappears. And that can disappear because perhaps you have some unexpected side effects in humans or some toxicity. So you can go a long way along the path and suddenly everything disappears. And I've been involved in several projects where you've got towards the finish line and then the project, the drug just disappears due to completely unpredictable circumstances. And the key thing really is to pick the right drug target in the first place. And that's where we are very well situated at Cardiff University to understand the pathophysiology of CNS and also cancer disorders. So that gives us a way of loading the dice in our favour, picking the right drug target. This is another way of showing the drug development process, going from target validation, so picking a target. So that's the person you saw in the earlier cartoon, sat at the desk, having a bright idea, going all the way to the right, phase 3, to the market. And what we've done at the Medicines Discovery Institute, and it's important to say that we've done this facilitated by the support of Cardiff University. And so this is unique within the UK. And by support, I mean the Cardiff University provided with the funding to underpin the infrastructure of the Medicines Discovery Institute. And that underpinning of the infrastructure, that underpinning of people, resources to do some early testing, is an investment, and that investment is hopefully going to pay off in big style with the launch of dry therapeutics. So this is the pipeline of drug targets that we've assembled. All the orange ones are CNS disorders. The purple one at the bottom is acute myeloid leukemia, which Loren will talk about in more detail. And at the bottom here, we see how at the left-hand side, we can fund projects based on things from the Medical Research Council, the Wellcome Trust, or charities. And as you progress to the right-hand side and things get more expensive, you run out of funding from people like the MRC, the Wellcome Trust, and you have to spin out a company or partner with Big Pharma. And in terms of the funds involved, these are very large sums of money at the right-hand side. So getting started in the first place is the cheap and cheerful end of the drug discovery process. But as we move into the clinical studies, so the costs really ramp up. In terms of psychiatric drug discovery, so myself and Simon, the directors of the MDI, have a passion for psychiatric drug discovery. We worked in companies where psychiatric drug discovery was deprioritized over the last 20 years or so, and we had an interest in drug targets. There was obviously a huge unmet need, so we set up the Drug Discovery Centre initially at the University of Sussex, and then more latterly at Cardiff University. And now the pendulum has swung back towards neuroscience. So this is just two presentations, two announcements of big deals that were struck at the end of 2023. So the pendulum has swung back in our direction towards psychiatric drug discovery. We've bundled up our three most advanced drug targets into drug therapeutics. Drug therapeutics has these three projects. We call them DT 101, 201, and 301. The key thing is that we have raised over $120 million of funding to fund these projects, and this is what we will be announcing in the very near future. So this will be the launch of drug therapeutics. So we've got $120 million to complete the phase 2 funding of our lead asset and 2 other projects through phase 1. These are the people involved. So just a slide to show that we are fully enabled in terms of the drug discovery aspects of drug therapeutics. We have funders from ICG, SV, Health Investors, etc. A world-leading team of scientific advisors. So that's drug therapeutics, which will launch in the very near future. And this just shows our pipeline of our three lead assets. The AMPA receptor, PAM, for major depressive disorder, will start phase 2 studies later in the year, and that's why we need a large amount of Series A funding. So that's a very broad overview of the Medicines Discovery Institute. Now, I want to introduce you to Loren, who is one of our stars of the Medicines Discovery Institute. So she joined as an MRes student in 2020. Obviously, this was during the time of the pandemic, but we were all hugely impressed by how independent and how capable she was. She worked on a project for Fragile X syndrome. So we were so impressed by her, we snapped her up and said, would you come and join us once your MRes finishes? And this has joined us in terms of a position that is funded by Cardiff University. And in that position, she conducted experiments that then led to a £2.3 million grant application to the MRC for acute myeloid lymphoma. And this is what she was talking about. And so she's been employed as part of that MRC research grant. I'll try and embarrass her a little bit. She's hugely enthusiastic, energetic person. She's independent, extremely thorough, questions her own work, great lab citizen, a great team player, really is a star of the MDI. And so with that, I'll hand over to Loren. So over to you, Loren.
Thank you, John. So as John mentioned, I've been working on this oncology project for acute myeloid leukemia, AML, which is an aggressive and fast-growing cancer of the white blood cells. It involves a buildup of abnormal cells in the bone marrow, which then interferes with normal blood cell production. So AML is the most common form of blood cancer in adults, accounting for about 25% of cases, particularly affecting those over 60 years old. Current treatments available for patients are high-dose chemotherapy, which is not always suitable for older patients. It can be quite harsh on the body. There's bone marrow or stem cell transplants. And in some cases, there's also CAR T cell therapy, which uses your own immune cells, modifies them to find and kill the cancer cells. But these treatment options are not always suitable or effective for all patients. And with a poor survival rate and increasing probability of relapse, there's a real need to find new and effective ways to help these patients. And next slide, please. So we are interested in a protein called CAT2A, which regulates the expression of genes associated with many different processes, such as cell growth, stem cell differentiation, blood cell production, metabolism, and activation of the immune system. But cancer cells can make use of regulators such as CAT2A to benefit their own survival and progression. And this was noted by researchers who showed there were high levels of CAT2A in many cancers, including AML. And they showed that by specifically targeting and stopping CAT2A activity, it then helped to stop the cancer cells growing and actually push them into a more matured cell state. And I'll explain this a little bit further later on. So on the next slide, please. So there are different approaches that we can take to effectively target a protein and stop its activity. You can use a small molecule to bind the target and inhibit its activity by preventing it from binding to anything else. Or you can use molecules called PROTACs, which are degraders, and these break it down and remove it completely. And this is what we wanted to do for CAT2A. So the way that PROTACs work is they hijack the body's natural protein removal and recycling system. So it essentially brings the target protein close to an enzyme called E3 ligase. This then flags the protein so it can be recognized for removal. Then the protein is collected and taken away to be broken down through the proteasome or the recycling center. And next slide, please. So going back to CAT2A's role in cancer cells, these abnormal blood cells become stuck in this second stage of development usually. They've been blocked from maturing into specific cell types, and so they stay in this uncontrollable state where they can then grow and build up. And CAT2A was shown to be involved in keeping these cells blocked. So the idea is that if we use this PROTAC system in AML to then take and break down CAT2A, it can then remove this blockage to help stop the cancer growing uncontrollably and even push them into a more mature cell state and eventually lead to cell death. And what makes this approach really exciting is that most cancer treatments target to kill the cancer cell. However, by targeting CAT2A, we're also addressing one of the underlying drivers that's helping to form this abnormal cell. And by removing this, we can then try and stop the cancer growing as well as kill it. And that's really important. So a lot of work has gone into this project, and we have explored the whole idea of CAT2A as a target. The chemists have developed a really good compound, a drug, that we can see effectively degrades the protein in AML cells. So as shown in this Western blot here, for the CAT2A bands, you can see they fade quite nicely. So this is showing there's degradation of the protein following this removal. As you add more drug to the cells, you can see that there's a sufficient drop in the growth curve. And whilst this has been really promising, we are seeing these good effects on the cancer cells from our drug. But what we need to understand next is how this would then be applied in a clinical setting. So translation-wise, what would the best dosing level and frequency be for patients, potential toxicities, and how we could then see these beneficial effects at a larger scale. And this is what we are working on now. Thank you for listening.
Thank you very much indeed, Lauren and John. Just wonderful to get a real flavour of team science at its best, and in the hands of drug hunters at the same time. So wonderful to learn a little bit more about the flavour of what you do. We're going to now move to the part of the evening where we take questions from the audience. And to kick things off, I'll start with a pre-submitted question before we move into the live questions. Just a reminder to the audience to think about questions that you might like to have answered, and we'll pull a selection of those to Lauren and John here this evening. So to the first question then, is Cardiff University or any university able to develop a medicine the whole way to market, or does it need to partner with industry? How is this process best achieved, and does it always work, or is it risky?
Yeah, so I'll take that. Thanks, Roger. It's hugely risky. A university by itself cannot develop a drug all the way to the market because of the huge expense and the risky nature of that process. So we are looking to take some of our assets, as we have done, to spin them out and get commercial funding. So this is venture funding, investors that want a return on their investment. So we've got 120 million in our Series A funding, and they'll be looking for a return on that investment. Now, it's very risky. Things could go horribly wrong very quickly, but hopefully they won't go horribly wrong. And if we are successful with our phase 2 studies, and then we could hopefully get some additional money to go to Series B funding to get through phase 3, or perhaps an IPO. Lots of other ways of getting this drug or these drugs to the market. But by itself, a university cannot fund a drug all the way onto the market. But actually, what Cardiff University has done, I think very uniquely within the UK, is provided underpinning funding. And that's not just a PhD student here and there. This is serious funding, serious lab refurbishment. We've got state-of-the-art labs. And so Cardiff University has been really enabling, and has had the vision and shared the vision that we have as directors of the MDI, that if you're going to do this kind of thing, you've got to do this seriously. You can't cut corners. You can't do it on the shoestring. You have to do it properly and provide resources. We have what we call the engine room of the MDI, which are Cardiff University funded personnel that do the kind of experiments that Lauren did initially to develop the data that will then underpin a grant application. And that is pretty unique in the UK. There are similar drug discovery institutes within Oxford, Cambridge, and London, the usual suspects, funded by Alzheimer's Research UK, for example, or the centres funded by Cancer Research UK. But as far as I'm aware, Cardiff University is the only one that provides serious underpinning funding, and so for that, it should be hugely proud, and we're hugely appreciative. Certainly.
Thanks, John. Yeah, comprehensive answer, and as you say, many steps on the journey through to the end point there. I'll move now to the live questions that have come in and pick one out. Here we go. What are the biggest barriers to getting drugs approved?
Getting them approved is, I guess, getting to the finish line, really. There's lots of things can go wrong. You can have unpredictable things, that's snakes and ladders analogy without the ladders. Lots of things can go wrong, things can disappear. You have to show that your compound is not only safe, but also efficacious. And that's a considerable challenge. A lot of the things that we have in clinical trials for psychiatry are old tools, ratings, scales where you have huge placebo effects. So we really do desperately need new ways of assessing the effects of a drug in a patient population, biomarker approaches. But we're in a catch-22 because regulators are used to seeing drug studies conducted with old rating scales, and so that's kind of what they expect. So you are locked into a cycle of tradition, really, which is not necessarily the best way forward particularly for new drugs. But yes, there's huge challenges.
So I guess, from the regulation point there, it's the conservatism that is making that custom and practice kick in, and also potentially making progress difficult perhaps through some of those practices.
Yeah, and I think it's fair to say that regulators don't necessarily concern themselves with the marketing of a drug. So they'll approve a drug, and if you can make a commercial success, great. If you don't make a commercial success of it, and there's lots of examples that of drugs that have made it to the market and not been a commercial success, well, the regulators don't care about that. They just want to make sure that the drug is safe and well tolerated and efficacious, and the marketing and how much money you make or don't make is none of their business to a certain extent.
Understood. Thank you very much for that. There's a quite specific question here that I'll pull in on the technical side, just to get some balance in. So bear with me as I work through this. With CAT2A drug targeted PROTAC inhibitor, how do you deliver drug to cancer cells specifically, or is this delivered systemically? If so, are normal tissues unaffected by CAT2A inhibition?
It's a good question. So it's delivered orally. Is that right, John?
Yep.
Yeah. And there's always the risk of affecting healthy cells. But I think that's things that we look at and we test. And it's with this higher expression of CAT2A in the cancer cells specifically, these are a lot more affected than healthy cells. I can't remember the second part of the question, sorry. What was that?
Is this, how do you deliver to the cancer cells specifically, or is this delivered systematically? And if so, are the normal tissues unaffected by the CAT2A inhibition?
Yeah, well.
Deeply technical. Sorry, it's putting you on the spot there, Loren. I don't know.
No, it's okay. I guess that is the big question: is how do we know it is only affecting the cancer and not the healthy cells? But yeah, that's what we look at. So,
and I think that it's a double-edged sword, because this is so novel and so innovative that we think it's a great idea, but until you actually test it for safety and tolerability, you don't really know. So there's a lot of empiricism involved, that you have a great hypothesis, and what Loren has described is a really innovative approach, this PROTAC approach, rather than rather than small molecules. And the PROTAC is a big old thing. It's a big
Huge.
molecule, and I'm amazed that these things aren't chomped up and metabolized really quickly, but they survive. They've got good pharmacokinetics, so that's absolutely amazing. But then the safety and tolerability is another question. That's one of those trapdoors that might open up once we start testing in safety and tolerability in animals, that it might just disappear. There might be some completely unpredictable toxicity that we've not foreseen.
And the bottom line is safety is so important in this area, isn't it? It's absolutely critical. Hence why the importance of the regulators in ensuring that for care of the public. Thank you both for explaining that. I'll move on now to a cost-benefit question. Is the cost of experimental therapy a consideration, given the cost-benefit and risk-benefit considerations in the UK and EU?
Yeah, so that's a good question. So when we got these projects funded by the MRC and the Wellcome Trust, we choose indications and disorders where there's a large unmet need. And so it's the medical need that is driving the funding of the projects. As we move into the commercial region, then particularly with the funding of the dry therapeutic, we have to then think about the commercial consideration, because the investors want a return on their investment. And so there's a little bit of a difference between getting projects funded for an unmet need versus a therapeutic indication where the investor can hopefully get a return on their investment. But with a lot of these drugs that we work on in the CNS, what we think is a good starting point from a disease point of view might turn out to be something completely different. And so that's what we get projects funded for a certain indication, and then in all probability they might end up in a completely different indication. And so our lead project, for example, was initially funded for schizophrenia, and has been more recently repositioned for depression because of a convincing emerging biology story that those kind of drugs are good for depression.
Thank you. Really interesting to unpick how the drugs can work in different ways inside the body. It's absolutely fascinating. Final question now coming up about what you've done in Cardiff. So this question is asking, you said the MDI in Cardiff is fairly unique. What did you do in Cardiff that makes it work so well?
Well, I think several things. So there's the vision of the university. We came to Cardiff University, and that required a lot of funding, labs refurbishing, like I say, a funding of the engine room of the MDI, and that takes quite a bold initiative from the university, because the university clearly wanted to get into a bit more of the translational side of things. So the university shared the vision that myself and Simon had in terms of translating science into patient impact. The other huge attraction of coming to Cardiff was the quality of the academic colleagues that we work with. So we can just have conversations, like I mentioned before, with the world-leading scientists in schizophrenia genetics or Alzheimer's genetics. We can just have conversation with them. What has been hugely impressive about coming to Cardiff is seems to be an absence of egos, so we can just have conversations with people. And it's really easy, really straightforward. We've got the infrastructure with people at the Kubrick, the Cardiff University Brain Research Imaging Centre. Again, that's been hugely enabling for our most advanced project. And I think it'd be safe to say that if we weren't at Cardiff University, that the drug therapeutics spin out would have not happened.
Thank you so much, Loren and John, for painting the picture of the work that you do, and also the wonderful environment that you've created and developed alongside the investment from Cardiff University. So fantastic. Thank you very much for your time this evening in bringing this to life, and also fielding questions from the audience. It's been really fascinating. Diolch yn fawr. I also want to thank the webinar audience for being with us this evening, for asking questions. We hope you found it interesting. If we didn't get a chance to answer your question, we will follow up with you directly to make sure that we provide a response. You will also receive an email in about a week or so. It will provide a link to the recording this evening, and also links to the university's future events diary. Information on how to get involved with Cardiff University, volunteering time to help with our students, or helping us to fundraise for such fascinating and fundamental research that can really make a difference, will be in the follow-up email after this event. So from myself, Loren and John, diolch yn fawr, thank you very much for your time this evening. And for the moment, it's goodbye from us.